research, different stage of career, and again, if you have any problem with connection or anything like that, let me know, or Diana, and we will situate you so you can join us every week. We have exciting next talk as well. But before I will turn to Emily for a formal introduction, I just will say two sentences about Jeremy and how he intersected with my career. So I finished my organic chemistry and biochemistry training and started working on vision with Paul Hargrave. And there were the 4 papers published by Jeremy as an MD PhD student from Stanford at that time. And if you haven't read those papers, you must. They are just really breaking ground in color vision. And they are in my drawer here among maybe 20 or so key papers in vision. And the beauty of that is not the technology, because of course that changed, but the intellect behind the study. And I think this is really something that I will make exam for every student if they know by heart those papers. So this is how I met through publication Jeremy, and since then he's my big hero in vision research. All right, that's I think enough. I see that Jeremy's ears are burning, so I will turn to Emily.
All right. Hi, everyone. Thank you for joining us for our seminar this morning. My name is Emily Tom, and I am a second-year MSTP student. It is my great pleasure to introduce our speaker, Dr. Jeremy Nathans from Johns Hopkins University. Dr. Nathans earned his PhD in biochemistry and MD from Stanford University. At Stanford, working with Dr. David Hugnes, Dr. Nathans isolated the opsin genes and showed that variations in these genes cause color blindness. After establishing his lab at Johns Hopkins as a Howard Hughes Medical Institute investigator, he and his team characterized gene mutations responsible for several retinal degenerative diseases, such as retinitis pigmentosa, Stargardt disease, and vitelliform macular dystrophy. Currently, his lab is interested in studying the frizzled family of cell surface receptors and their roles in the Wnt signaling pathway in neural and vascular development. He and his lab have engineered novel genetic tools and techniques, such as the ability to visualize and manipulate single mutant cells in mice. Dr. Nathans' contributions to science have not only changed our understanding of retinal molecular biology and diseases, but also how humans see the world. He has been elected to the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Sciences. He serves on multiple advisory boards and as an editorial board member of Proceedings of the National Academy of Sciences, and has been recognized with numerous teaching awards at Johns Hopkins. Most recently, he was awarded the Benjamin Franklin Medal in Life Sciences. Thank you again for joining us, and without further ado, Dr. Jeremy Nathans.
Thank you, Emily, and thank you, Chris, for that very generous introduction. And also thanks to Diana for running the show here. I'm going to take over the screen, and I hope I do this right, and then we'll proceed. Okay, I think we're good. So today I want to tell you about work we've been doing recently on the genetics and biochemistry of CNS vascular development and disease. And before we talk about the experiments, I want to mention...
You're not sharing.
Say it again?
You're not sharing the screen, do you?
Uh-oh. Oops, hold it. I screwed up already. Oh, I didn't hit screen share. Holy cow. Diana did tell me how to do this.
We all learn to listen to her.
She told me how to do it right. Okay. Yeah, now
that's working. All right, great. Thank you.
Perfect. Okay, let's run that play again. So what I want to tell you about today is work we've been doing on molecular genetics of CNS vascular development and disease. And before we get to the data, let me just mention some of the people who've done the work. and in particular from my lab, Chris Cho, an MD PhD student. Can you see the cursor, by the way? Yeah? No?
Yes. Yes, we can.
Chris Cho and Amir Ratner and Mark Sabah, Phil Smallwood, Yanshu Wang, and Xin Yi and Yulian Zhu are the main movers on this. And really the project, I think it's fair to say, stems from our fascination with vascular biology, shown here in Vesalius's drawing from the 16th century, and in particular the part of the vasculature that supplies the brain, spinal cord, and retina. And as you'll see, there's a series of specialized both properties and signaling systems for that vasculature. And the signaling system that we have focused on is canonical Wnt signaling. We'll also call it beta-catenin signaling interchangeably. And I've just shown a simplified version of it here, at least as it occurs at the membrane of a cell. So the horizontal black lines are the parallel lipid bilayer outer and inner leaflets. And there is a family of receptors called frizzled with 7 transmembrane segments. There is a family of ligands called Wnt. There are 19 Wnts in mammals, and there are 10 frizzleds in mammals. There's a co-receptor, LRP5 or 6, virtually identical single-span transmembrane proteins. And together, these proteins, when part of a signaling complex, they activate a stabilization of beta-catenin, a protein which is normally in the cytosol at rather low concentration. Upon stabilization, it migrates to the nucleus and controls transcription. So I'm going to divide the talk into 4 parts. The first is about Noren and frizzled 4 signaling in retinal vascular development. And the story begins about a century ago with this ophthalmologist, Gordon Nori. He is the person who first described the disease that now bears his name, Nori disease. It was not really fully described, I think it's fair to say, until Meta Warburg, about 50 years later, described in detail the clinical presentation and also the genetics of a number of patients and their families. And I will just show you one pedigree from her paper. This is a 1961, I believe, paper. And you can see in this pedigree, every square is a male, every circle is a female, that the individuals who are affected are only males, and they are related to each other in a pattern that is consistent with X-linked inheritance. And in fact, the disease is X-linked. And clinically, it presents as congenital blindness. And if you look in the eye, and this is now clearer now that we have mouse models, the basic problem is that the retina is severely hypovascularized. There are not enough blood vessels, and that leads to destruction, gliosis, and loss of vision. Now, more recently, it's become apparent that there are milder related disorders, hypovascularization disorders in the retina, and they fall under this umbrella term of familial exudated vitro retinopathy. They are inherited, hence the word familial. And here's an example on the left of a normal fundus image, and you can see the optic disc on the left side, and here's the fovea in the center. And on the right, an image of someone who has fever, we call it FEVR, for familial exudated vitro retinopathy. And this individual has a region of retina which is hypovascularized. It has led to a scarring response in that region and retraction of the retina. So you can see the retinal fold here. There's a detachment of the retina in this right side. And of course, that's not good for vision. Now, it became clear in the early 2000s, thanks to the work of Johann Robitaille, a Canadian ophthalmologist, that in one subset of fever patients, there is heterozygosity for mutations in the frizzled 4 gene, one of the 10 frizzled receptors. And we and others had shown some years earlier that these are the receptors for the Wnt proteins. And so that immediately linked Wnt signaling with vascularization in the retina. And at essentially the same time, we and Wolfgang Berger's group showed that the frizzled 4 knockout mouse, now this is the complete loss of function, not the partial loss of function that you see in human fever patients, but the complete loss of function frizzled 4 knockout mouse or a Noren knockout mouse. And here, I'll just say the Nori disease gene is called NDP, and the protein is called Noren. NDP stands for Nori disease protein. But if you knock out either of those in a mouse, you get essentially the same phenotype, which is a failure to form the normal capillaries. So this is a cross-section of a retina, and on the left we see the normal 3 tiers of capillaries, one tier between the outer nuclear layer and the inner nuclear layer, a second tier on the inner side of the inner nuclear layer, and the third tier on the vitreal face of the retina. And you can see that the vessels are somewhat hypertrophied. They have started to invade the retina, but really haven't gotten very far, and they certainly haven't made capillaries. And this essential identity between the frizzled 4 knockout phenotype and the Nori disease knockout phenotype, at least in mice, suggested to us that maybe frizzled 4 was in fact a receptor for the Noren protein. And I should mention that at this point, the Noren protein, which had been isolated by others by a positional cloning method, it was easier to clone because of its X linkage than the typical gene in those early days. That protein clearly looked like a ligand. It's actually a divergent member of the TGF-beta superfamily. So it was a ligand looking for a receptor. Frizzled 4 is a receptor, although at this point we knew that it was a Wnt receptor, and so it's not as if it was looking for a ligand. But this coincidence in phenotype seemed too impressive for the two not to have anything to do with each other, and in fact that guess was correct. Noren is a ligand for frizzled 4. It's a very good ligand for frizzled 4. You can show this in a luciferase assay. So this is a transfected cell system in which a luciferase reporter reports on canonical Wnt signaling, and you can see that. Oops, sorry, my little arrow is freezing here. Oh, there we go. Okay, you can see that if we transfect with frizzled 4 and the co-receptor LRP5, but we leave out the Noren, there's very little signaling. When we have all 3 present, there's a lot of signal, and the same is true for the LRP6 co-receptor. And not only does Noren induce signaling in transfected cells, but Noren also binds to the frizzled 4 receptor with nanomolar affinity. So here's just an example where we've produced the extracellular domain of frizzled 4 as an IgG fusion, and then done the binding curve, and it's a several nanomolar affinity binding. And here we've just done the binding to intact cells expressing full-length frizzled 4, and we've got controls down at the bottom that don't bind. Now, not only does Noren bind frizzled 4 tightly, but it also binds it very specifically. So here's an experiment where we have transfected cells with the extracellular domains of each of the 10 frizzled. So frizzled 1 through 8, and then 9 and 10, plus a series of frizzled-related proteins. These are really decoy receptors, but they have homology to frizzleds. And so we've transfected cells with constructs that display each of these regions on the surface of cells. And that's shown on the left because they're MYC tagged, and we can see the MYC epitope staining. And then in the central column of each of these, we are probing with Noren, and in the rightmost column with a Wnt8 probe. Just happens to be a Wnt that's especially well behaved, so easy to use as a probe. You can see that the Wnt binds to 4 of the frizzleds: frizzled 4, frizzled 5, frizzled 7, and frizzled 8, but not in the others. But Noren is exquisitely specific for frizzled 4. It binds frizzled 4, but it does not bind the other frizzleds. So Noren is a highly specific ligand for this one frizzled. And now the crystal structure of the Noren frizzled 4 ligand binding domain has been determined. This was done in Yvonne Cheng's lab about 5 years ago. Noren is a dimer. It's shown in pink here. It's a disulfide linked dimer like all TGF-beta family members. And here on left and right in blue are the ligand binding domains of the frizzled 4 receptor. Now, all of that that I've told you so far is in cell culture. Those experiments are in cell culture or in vitro. And you might be wondering, does all this hold up in vivo? Let me just show you one piece of data that says that it does. So here's an experiment. If we just look at the bottom row here, this will tell the tale. This is an experiment in which we have rescued the Noren knockout phenotype by artificially activating canonical Wnt signaling specifically in vascular endothelial cells. So let's look on the left first. What you're seeing in this bottom row, this is an en-face view of the retina. So we flat mounted a retina. We're looking down on it from the top. And what we've visualized are just the blood vessels, and they've been color-coded based on their depth. So we're seeing three different colors, red, green, and blue, which correspond to the three different layers of retinal vessels. And then in the middle panel, we're seeing the Noren knockout phenotype, which shows these little balls of cells. These are endothelial cells that have gotten a little bit of the way in the retina, but they really haven't gotten very far. They haven't made capillaries, and that's the knockout phenotype. Now, if we use the usual mouse genetic tricks and we just activate beta-catenin, we stabilize beta-catenin. I'll mention this is a trick in which one exon of the beta-catenin gene, which is a very tiny exon, it only encodes a handful of amino acids, includes a phosphorylation site which controls the degradation of beta-catenin. So if you excise that exon, then beta-catenin is far more stable than it would normally be. And it turns out, this is a gift from the gods, that exon is a multiple of 3 nucleotides. So when you cut it out, you do not introduce a frame shift. So that's what we've done here. We've got that beta-catenin conditional knockout. It's exon 3. It's abbreviated CTNNB1. That's the beta-catenin gene. Flex 3, the floxed exon 3. And we recombine it with a CRE-ER specifically expressed in vascular endothelial cells. So basically, we're bypassing any defects at the plasma membrane. It doesn't matter whether the receptor or ligand are there or not. We're just stabilizing beta-catenin intracellularly, and you can see it fully rescues the defect. So this is basically what is wrong in people with Nori disease and fever. They're missing canonical Wnt signaling in their vascular endothelial cells. Okay, now let's expand the discussion and talk about vascularization in the brain and spinal cord. And this, of course, is the other part of the CNS. The retina is part of the central nervous system, but only a little part of it. And it turns out, I'll just spill the beans here, that this same signaling system, although it uses some other partially redundant players, but the same basic signaling system is used throughout the central nervous system. And it is also, as we'll see in a minute, used not just in development, but also in adulthood. So here is an example of a mouse embryo at essentially mid-gestation, embryonic day 11.5, a little after mid-gestation. We're looking down at the top. This is the neural tube. And in a normal embryo, we can see the somites left and right, and we can see in green the blood vessels invading the neural tube, the growing neural tube. On the right, we have an embryo in which we have eliminated the beta-catenin gene from all vascular endothelial cells. Now, amazingly, except for the central nervous system, the embryo is completely fine. The heart is fine, the placenta is fine, the somites are fine. But in the central nervous system, there is essentially no invasion of vasculature. This white stuff that you see is actually leaking intravascular contents. It stains a little bit in this technique, but there are no capillaries that have entered here and formed. So something is special about canonical Wnt signaling and central nervous system angiogenesis. And it's not just the retina, it's also the rest of the CNS, but of course in development, early development. Okay, now let's take a slight detour, and I'll connect it in just a minute to show you where this path took us. So the detour takes us to the oncology department here at Johns Hopkins, and to the lab of Ken Kinsler and Bert Vogelstein, shown on the right here, two of the greatest scientists in the oncology field. In fact, Bert Vogelstein is the most highly cited scientist on the planet. Ken Kinsler is not far behind. They together run a research lab here, and about 20 years ago, Brad St. Croix, at the time a postdoc in the lab, did a very important experiment. He asked whether there was differences in gene expression in tumor endothelial cells, that is the cells that line the blood vessels, in tumors relative to normal tissue. And the answer was yes, there are differences in gene expression. There are very few of them. It's remarkably specific. And a series of membrane proteins, or the messages that code for those membrane proteins, came out of this search, and they called them TEMs, tumor endothelial markers. And there were exactly 8 of them, TEM 1, 2, 3, 4, 5, 6, 7, 8. TEM5, which I'm going to tell you about, was a known sequence. It corresponded to what was then called an orphan G protein coupled receptor, GPR124. It wasn't really known to be a G protein coupled receptor. It just had, by computer analysis, 7 putative hydrophobic segments, presumably transmembrane segments, so it looked like a G protein coupled receptor. Everyone sort of assumed it was. What Brad St. Croix and his colleagues showed was that by in situ hybridization, the TEMs, and here TEM5 is on the right, really conformed to that pattern that came out of that initial, turns out it was a SAGE study, a method that was used to quantify message abundances. Now, here it's refined by in situ hybridization, and you can see in red, which is the in situ hybridization signal, that the blood vessels in this human colon tumor sample, colon cancer sample, are highly enriched for TEM5, but it's essentially undetectable in normal colonic epithelium, the surface epithelium of a normal colon. Now, about 8 years later, several groups reported the knockout phenotype of GPR124, or TEM5, essentially simultaneously, and what they saw was quite spectacular, and that is that it had a defect in angiogenesis in the brain. And that's shown here. We've just reproduced that data with the line that we got from the Jackson laboratories. Here is a section shown by this white line through the head of an embryonic mouse. In green are the blood vessels, and here, if we just look at higher power, here's the cortex with lots of capillaries in it. Here's the ganglionic eminences, also lots of capillaries. And in a GPR124 knockout mouse shown on the bottom line, the bottom series of images, we see essentially no capillaries in the cortex, and it's somewhat thinner as a result, and essentially no capillaries in the ganglionic eminences, and they're starting to necrose. Now, I'll just back up a second. Now that really struck us, because at this point, not only did we know that canonical Wnt signaling was critical for retinal vascularization from our work on Nori disease, but the group of Andy McMahon and also the group of Ben Barris had shown at this time that canonical Wnt signaling, in particular mediated by 2 Wnts called Wnt7A and Wnt7B, very closely related Wnts, was critical for vascularization in the brain and spinal cord. And in fact, the pattern of defects shown by Wnt7A, Wnt7B double knockouts, and I should say they're somewhat redundant, was essentially identical to this GPR124 phenotype. This, however, this identity seemed to escape the notice of those in the field, because the groups that published on this knockout basically concluded that this was a G protein coupled receptor with a novel signaling system that was controlling vascular development. So that was obviously an exciting possibility. But what Yulian Zhu, a grad student in my lab, and I thought was, this has got to be related to Wnt signaling. And so we simply asked, is GPR124 somehow involved in the efficiency of Wnt signaling, and is it specific for Wnt7A and Wnt7B, the 2 Wnts which McMahon and Barris had shown are critical for CNS vascularization? And so Yulian did a very simple experiment. She compared the level of signaling activity in this transfected cell system when the cells get each of the 19 Wnts, or Noren, and GPR124 and the co-receptor. And here I should say we don't have to add the frizzled, because these cells, as it turns out, express low levels of essentially every frizzled. So it's already there. The frizzle's already there. If we compare that signal in the presence of those components to the signal we get with just Wnt and an empty vector, PRK5, instead of GPR124 and the co-receptor LRP5, what's the intensity of the signal that we get in those 2 situations? And we look at the ratio of those 2 signals. And here we're plotting it, so that's on the vertical axis. And on the horizontal axis, we're plotting the actual signal, the Wnt plus GPR124 plus LRP5, which spans an enormous range. That's why it's a log plot. Some Wnt's, as it turns out, work extremely well, like Wnt1 and Wnt3A. Others work rather poorly. But that's okay. They lie along a wide range on the horizontal axis. The question is, what's the ratio? And for, as you can see, for almost all of the Wnt's, the ratio is very close to 1. That is, there's no effect of adding GPR124 on signaling in this cell culture system. but that's not true of Wnt7a and Wnt7b. They are enhanced by GPR124. And these are exactly the two Wnts out of all 19 that had been shown to be involved in CNS vascularization. Well, to compress a very long story and make it mercifully short, GPR124 is some sort of, we call it a co-activator, some sort of component of the signaling complex at the plasma membrane. And there's yet another one, first discovered in zebrafish, but we've been working on it from the beginning, called REC, which is a multi-domain GPI-anchored protein. And together, REC and GPR124 and the ligand, 7A, Wnt7a or 7b, and frizzled receptor and LRP6 or 5 co-receptor is the complete, we think, signaling complex. And just to give you an idea of how specific all of this is when you put it together, here we're comparing for a subset of the Wnts, all the other Wnts look pretty much the same, the level of signal that you get when you transfect them into cells, again, the luciferase assay, and add either just the empty vector, PRK5, in addition to the Wnt ligand, or GPR124, or REC, this other co-activator, or REC plus GPR in blue. And I think you can see for essentially all the Wnts except 7A and 7B, it really doesn't matter which of those pieces you have. The levels are different for different Wnts, but the levels don't change very much depending on the different pieces, except 7A and 7B show this huge enhancement, and it's quite synergistic with REC and GPR124. Having both of them is critical, although you get a little bit of a boost with just one or the other. Actually, I should say the reason you get a little boost with one or the other is because these cells that we use for the reporter line actually make a little bit of REC and a little bit of GPR124, so you get a little bit of synergy. Now, not only is the activation incredibly specific to those 2 out of 19 Wnts, but binding in this complex is specific. And we've demonstrated that. This was work of Chris Cho, an MD PhD student, by making alkalinephosphatase fusions that can be used as probes, and in this case using the front end of REC. So these are a series of domains that we call the CC domains. There are 5 of them, CC1 to 5. That's fused to the alkalinephosphatase probe. And it's used to probe transfected cells, which are shown here. The cells are very tiny in this image. It's a low power image. And the cells have been transfected with GPR124 and frizzled 4, and each of the Wnts individually, or Noren, and then incubated with this soluble alkaline phosphatase probe. And what you can see is the soluble probe can form a co-complex with these other pieces on the cell surface, but only when 7A and 7B are present as the ligand, not for any of the other Wnts. And the same is true on the right side, where we've just used a piece of GPR124, again, the extracellular region that's the business end. It's 2 domains, a leucine-rich repeat domain and an IG domain. That is now used as the probe. And the other pieces, in this case, REC and frizzled 4, and each of the various Wnts or Noren are transfected. And again, you can see 7A and 7B are the ones that are required for that complex. So here's a system that is exquisitely ligand specific. And we think actually this is essentially tuning the specificity. Having GPR124 and REC is tuning the specificity to Wnt7a and Wnt7b. Now again, like that first part of the talk, this is all in vitro, it's in cell culture. You might be wondering, is this really true in vivo? That is GPR124 really activating canonical Wnt signaling? And if that's true, we can play the same trick that we played in the retina, which is we can try to rescue the mutant phenotype by stabilizing beta-catenin specifically in vascular endothelial cells. So that's what we've done here. You've seen the upper 2 rows before. That's the topmost row is the wild-type mouse. We can just look at the cortex, which tells the story. The wild-type mouse has capillaries in its cortex. The GPR124 mouse lacks capillaries in the cerebral cortex. And here is a wild-type mouse in the 3rd row, in which we have simply stabilized beta-catenin in the vascular endothelial cells. There isn't much of an effect. It doesn't seem to be much difference whether there's a normal amount of beta-catenin or an excess of beta-catenin. But the bottom row is the important one. This is the mutant mouse where we've stabilized beta-catenin. And there's a substantial rescue, and likewise for the ganglionic eminences. It's not a perfect rescue, I would say. And I think the reason is that this had to be done as a Cree-ER experiment with tamoxifen injection. And I think the activation of the recombination is not 100% efficient and probably not quite early enough to get a complete rescue. But basically, the story's right. It's canonical Wnt signaling. And actually, I'll show you another piece of data in favor of that conclusion, and that starts here. And that is that we had known for some years that Noren is in fact present in the brain and spinal cord in early embryos. We didn't see an obvious phenotype in the Noren knockout mouse, so we were wondering what it's doing there. And of course, one possibility is that what it's doing is partially redundant with what Wnt7a and Wnt7b are doing. And that turns out to be true, and it's especially obvious in the hindbrain. And sections here are through the hindbrain, just to show you this. So now it's a black and white image, and the black lines are the blood vessels, the little capillaries. And what we're seeing is that in a wild type animal, the capillaries are normal in the hindbrain. In a Noren knockout animal, the same is true. In a GPR124 knockout, it's not really perfect. There's some disorganization and hypovascularization. And if we look at a heterozygous with Noren, it gets a little worse. But where it really gets bad is if we get rid of GPR124 and we also get rid of Noren in the same animal, then there's basically no vascularization. You just get these little balls of endothelial cells, just like we saw in the retina. So these two systems, the GPR124 REC Wnt7a, Wnt7b system and the Noren system, are essentially redundant in the hindbrain. And I should say they're redundant in some other places. And so this led to the following experiment. This is one in which we're rescuing the GPR124 phenotype simply by flooding the brain with Noren, this different ligand, which doesn't use GPR124 or REC. I should have mentioned that. So those two co-activator proteins are exquisitely specific. I guess we did sort of mention that. They're exquisitely specific for Wnt7a and Wnt7b. They're irrelevant for Noren signaling. So again, you've seen the top two rows before, the wild type mouse, the GPR124 knockout mouse, and now we're overexpressing Noren everywhere in the CNS, and there's basically no effect. It's just fine to have extra Noren on board. But if we overexpress it in a GPR124 knockout mouse, it fully rescues the phenotype. So again, all we have to do is activate canonical Wnt signaling in the vascular endothelial cells, and the blood vessels will grow just fine. And here I'll just add, I don't want to belabor this too much. We've done the same thing basically with REC. In this case, we did a lot of structure function analysis, and we identified two amino acids that are critical for REC's interaction with and signaling in response to Wnt7a and Wnt7b. It happens to be proline 256 and a tryptophan 261. So we used CRISPR to make exactly those mutations in the mouse germline. So we have this double amino acid substitution mouse. And when we look at the phenotype, it's got this spectacular angiogenesis defect phenotype. If we just look at the left set of panels, here's the wild type mouse. We don't have to worry about the staining. Basically, what we're visualizing are blood vessels with various stains. The blood vessels are pretty much everywhere in the CNS. And in the lower panel, what we see is the effect of this REC allele. It's over a second allele, which is milder. And what we see is essentially a complete absence of vascularization in the cortex and in the ganglionic eminences. So both of these components are required in vivo. And now let me just say something about the structure of Wnt's and frizzleds. There's a bit of a paradox in this field. Chris Garcia, an amazing structural biologist at Stanford, solved the co-crystal structure of the ligand binding domain of frizzled. It happened to be the frizzled 8. We call it the cysteine-rich domain, CRD, which is in blue, bound to a Wnt. It's a Xenopus Wnt 8 in pink. And what he saw was a remarkable structure. The Wnt in pink is holding on to the blue target with just one thumb and one finger. It's like a fist, but it's like a fist holding a hand holding a hot potato. It doesn't want to touch it any more than it has to. That is, the two are touching, just barely touching each other. And the regions that are in contact, I might note, are generally comprised of fairly conserved amino acids. So this structure suggested that the interaction between Wnt and frizzled was quite unusual for ligands and receptors. It involved very little surface. I should say that most of the surface area actually is due to this lipid residue, this palmitate, which is covalently linked to the Wnt and sits in a groove in the frizzled cysteine-rich domain. But that's a completely conserved feature across all Wnts, like many of the contacting amino acids. And so this crystal structure really led to a paradox. So where's the specificity between Wnts and frizzleds? And we know there is specificity. It's not absolute. There's some redundancy, as we saw with the binding experiment about 20 slides ago. But there is also a lot of specificity. The question is, where does it come from? And we think part of it may be coming from these auxiliary proteins, like GPR124 and REC, which are basically choosing the right Wnt and bringing it to the right frizzled receptor. Okay, now the third topic, the blood-brain barrier and beta-catenin signaling and barrier plasticity. The blood-brain barrier has been around for a long time. It was discovered over a century ago by Paul Ehrlich and his two students, Max Lewandowski and Edwin Goldman. And it basically consists of a series of specializations to the capillaries in the central nervous system. So this is the spinal cord, the brain, and the retina, which are diagrammed here. This particular slide is in German. I guess it's appropriate because the three discoverers are German, but it's the best one I could steal from the web. And you can sort of figure out the words anyway, like astrocyte here and endothelial cell. But the bottom line is that the endothelial cells that line brain capillaries lack passive diffusional capability that capillaries have elsewhere in the body, for example, in the liver or the kidney or the lungs or the muscle. Here, there are tight junctions. There are no fenestrations. These are little holes that allow small molecules to diffuse. There are, of course, because there's no free diffusion, there are transporters to actively transport essentially everything that has to go from the lumen of the capillary. So this would be all of the amino acids, it would be glucose, anything else that the brain needs. All of that has to be actively transported one molecule at a time, and other specializations as well. So it's a huge program of gene expression that distinguishes capillaries in the central nervous system from capillaries everywhere else in the body. And it turns out that this barrier is of profound significance in medicine. It is compromised in a wide variety of neurologic disorders and also, I should say, retinal disorders. Here's just a small list from the neurology world. In stroke, multiple sclerosis, CNS infections, traumatic brain injury, tumors, epilepsy, Alzheimer's disease, the list goes on. There is loss of barrier integrity. And it turns out that the barrier is under the control of canonical Wnt signaling. So canonical Wnt signaling, as we've seen so far, is critical for development of the vasculature in the brain and retina. That's an early function, but the system continues to be active. The ligands continue to be made by the brain parenchymal cells, neurons, but even more so glia. And the receptors continue to be expressed on the vascular cells. And so throughout life, this system is signaling. It's signaling right now in your brain and my brain and retinas. And it maintains the integrity of the CNS vasculature. And here's just an example of that. And also this demonstrates the redundancy of the GPR124 and REC and Wnt7A, Wnt7B systems with the Noren system. So if we make individual knockouts, for example, we've knocked out Noren here, and we have a heterozygous for GPR124 over here, the brain is not leaky. Then I should say the red stuff, the reddish purple material is intravascular dye that we are using to monitor the leakiness of the blood vessels. Now, there's a lot of leakage in the ventricles. That's where the cerebrospinal fluid is made, and that's appropriate because the vessels that make the CSF, this is the choroid plexus, those are leaky. But elsewhere in the brain, for example, the cortex, the blood vessels have this barrier, and so there's no leakage. Well, this is fine in the wild type, and it's fine in the single mutants. But if you make a double mutant of both GPR124 and Noren, and this is done, of course, you can't do it from the very beginning because you have a dead embryo. But if you let the animal develop normally, and then in the adult, you have a CRE-ER system, a drug-controlled CRE, and then you give the drug to the mouse, and then activate recombination to eliminate the one remaining GPR124 conditional allele, then you get leakage pretty much everywhere. So on the upper right, I'm trying to get my cursor to move here, but there we go. Okay. In the upper right, we see this diffuse leakage of the intravascular marker, this red marker, everywhere in the brain because the canonical Wnt signaling system is now down and the capillaries are losing their barrier function. And we can see this also by immunostaining. And I'll show you this same pair of markers in some future slides. Green is a tight junction marker, it's Claudin 5. It's supposed to be there. And purple is PLVAP, a marker of permeable vasculature. It is not supposed to be there in the brain and retina. And you can see that in the case of the normal cortex, the vessels are all green as they should be, and they're not leaky. And in the case of the cortex where we've eliminated both the Wnt7a, Wnt7b system and the Noren system, the vessels have flipped their identity to a leaky state. And here's just to show the sort of plasticity of this system. That last experiment kind of showed it, but I just want to show it here again in an experiment where we've done the recombination with a very low drug concentration. So this is tamoxifen, the drug that activates the CRE protein. We can titrate that drug down to just recombine, for example, 1% of the capillary endothelial cells. And when we do that, we see that individual capillary endothelial cells, now the PLVAP is in red, have flipped their identity. So they should be green, as shown up here, but now they're flipped to red. And this individual cell, this is a single capillary endothelial cell, is now a site of leakage of this intravascular material. Here I'm afraid I'm confusing you because here it's blue this time instead of pink. But this is the same stuff leaking out into the parenchyma. You can see this little puff of blue dye leaking into the parenchyma. And the same is true at these other locations here. Wherever there's a capillary endothelial cell that's lost its Wnt signaling, it now has flipped its identity to the leaky state, and the capillary there is leaky. Okay, so one final topic, circumventricular organs. Let me just check the time here. Yep, I think we're good. Okay, so in the brain, you know, we talk about the blood-brain barrier, and that certainly applies to the vast majority of capillaries in the brain. But there are a small number of places, they're right along the midline, where there is a highly permeable capillary bed. And these are called circumventricular organs because they're near the ventricles in essentially, well, not every case, but most cases. And they do things that their role is to sense, in several cases, the contents of the blood. So it makes sense that they have highly permeable capillaries. So, for example, the subfornical organ is this tiny little bit of nervous system tissue which has a highly permeable capillary bed. And the SFO, as we call it, the subfornical organ, the SFO's job is to monitor the serum sodium concentration. If sodium goes up, that means you need to drink. And in fact, when you activate neurons in the SFO, they connect to the thirst center, and that causes the organism to seek water. If you look at the area postrema, just under the cerebellum, that is a region which detects toxins that are circulating in blood. Under natural conditions, when you ingest a toxin, it's because you've eaten it. You've eaten, say, a toxic mushroom. It's in your stomach. You've got a little bit of time to vomit and get rid of it. And that's exactly what the area postrema does. So the area postrema, when it's activated, causes the animal to vomit. It's actually a pretty big deal. In the wild, we'd probably be dead without it. It turns out it's quite important clinically because the reason that cancer patients get nauseous from chemotherapy, from cancer chemotherapy, is because the toxicity of the chemotherapy is detected. The toxic properties are detected by the area postrema, and that makes them nauseous. Now, I'll just mention one other place, the median eminence and the posterior pituitary. These are highly permeable, not because they're sensing what's going on in the blood supply, but because they're secreting peptides into the blood supply. In the case of the median eminence, it's acting on the posterior pituitary, the hypothalamic releasing factors. And in the case of the posterior pituitary, it's acting on the anterior pituitary, the hormones that cause release of the anterior pituitary hormones. So we were wondering, How is it possible that these regions of the brain have this distinctive vascular permeability? And does it have anything to do with canonical Wnt signaling? Like, is canonical Wnt signaling turned off in the circumventricular organs? And I'll tell you right now, that's exactly true. Canonical Wnt signaling is turned off. And if you turn it on, they will assume a blood-brain barrier characteristic. So here's just an example of this with the area postrema, which is shown here. Here are the capillaries within it. They're expressing this purple marker, PLVAP, because they're highly permeable. Around them are green capillaries in the normal blood-brain barrier competent part of the brain, and that's the green in this case is GLUT1, the glutamate transporter, which of course all of the blood-brain barrier capillaries need. And over here on the right, I'm just showing you the choroid plexus, the part that makes the cerebrospinal fluid. So if we look at leakage, we see that the choroid plexus is highly leaky, no surprise, because it has to make CSF. The area postrema is highly leaky because it has to detect toxins. But the other parts of the brain, a little bit of the cerebellum here, brainstem down here, are not leaky. Now, if we just play our usual trick and stabilize beta-catenin artificially in vascular endothelial cells, what we see is we've converted the area postrema to a blood-brain barrier state. Here it is. It's all green and not purple. And here's the same thing with the subfornical organ. As I said, this is tiny. It's just a little ditzel here, and it just has a few capillaries in it. This is a coronal view. Here it is in the wild type. It has lots of PLVAP, the leaky marker, and it expresses very little of the capillary cells, make very little of this LEF1 transcription factor, which is in the nucleus of all of the capillaries in the adjacent brain region. So these are nuclei of capillary cells. And now if we artificially stabilize beta-catenin, we see that we've changed, that's on the right here, we've changed the subfornical organ's capillaries from purple to green. Now they make Claudin 5 and they're tight, they're barrier competent. And we've turned on this LEF1 transcription factor, which is a direct target of Wnt signaling. It also actually happens to be a participant in Wnt signaling, so it's kind of a positive feedback loop here. But we've turned on LEF1 in these capillaries. I just want to look at the area postrema in a little more detail to give you a sense of how exquisitely precise this is anatomically. So first, on the left side here is a view looking down on the brain, and we're looking at the capillaries in the area postrema. They're in green. I'm sorry, I keep switching the colors on you. So, but the labels are on the left. But the green here, and I apologize to Don Zach, who's colorblind. This is green and red. I shouldn't have done this. Sorry, Don. The green is PLVAP, the marker of permeable capillaries, and that's in the area postrema. And then I think you can see that in the blood vessels immediately around it, including the ones that feed into these capillaries, but in the brainstem region adjacent to it, the blood vessels are blood-brain barrier competent. They're red. They make the GLUT1 transporter. And amazingly, as shown by these arrows, you can see this transition from red to green, which is incredibly sharp. It's a single cell junction here. So one capillary endothelial cell is red, makes GLUT1. The one right next door is green. It makes PLVAP. And it's all or none. These cells know what state they want to be in, and they're not beating around the bush. They're either in one state or the other, nothing in between. And in this case, we have some inkling of what's going on. If we in situ hybridize, now this is a coronal section, a different plane of section, through the area postrema, and we probe with WIF1. This is a soluble Wnt inhibitory protein that we discovered about 20 years ago. It binds essentially all Wnts and basically soaks them up. So it's an all-purpose inhibitor of Wnt signaling. What we see is it has this amazing expression. It's expressed exactly in the area postrema cells. We're not exactly sure which cells they are. They might be glia. That requires a single-cell RNA-seq analysis. Now, just to give you one piece of data that shows that WIF1 isn't just there, but it's actually doing something, although not doing everything, I would say. If we knock out WIF1 and we look at the state of the area postrema capillaries, what we see is that unlike the wild type, which is shown on the left, where all the capillaries in the area postrema are, I say almost all, are PLVAP positive, that is, they're purple, and there's essentially no green capillaries on the left, or very, very few. If we get rid of WIF1, there are quite a few more capillaries which have switched from purple to green. We don't know why they all haven't switched. Probably there are other inhibitory mechanisms, and there's some sort of thresholding effect. By getting rid of WIF1, we've probably brought the level of Wnt signaling down, but it's not zero. And so there are plenty of capillaries which are still purple and still have the high permeability state. And there's some other players here that remain to be discovered. And finally, I'll just mention the last thing that we're doing. This is work from Mark Sabah, who was an MD PhD student in the lab, now just graduated. Mark has done an analysis at the level of the whole genome of what is going on in the different types of capillary endothelial cells in different organs and in different regions of the brain. And we're interested in basically looking at the genome and asking what's the sort of the global regulatory landscape for genes involved in blood-brain barrier formation. And here's just an example of the kind of approach we've taken. So we've purified vascular endothelial cells from brain, from liver, from lung, and from kidney. And for each of those sets of cells, we've determined the expression of all the genes with RNA-seq. And so here's an example of SLC2A1, which is the glucose transporter. And you can see it is expressed specifically in the brain. So for each one, we have independent replicates. So this gene is expressed in brain capillaries, but not in liver, lung, or kidney capillaries. If we look at accessible chromatin with the ATAC method, we see that all of them have accessibility near the promoter site, essentially at the promoter site, but only the brain capillary endothelial cells have regions of accessibility further upstream. These are presumptive enhancers. And when we look, and this was with Joe Ecker's lab at the Salk Institute, at whole genome methylation profiles, so methyl C-seq, and we look at methylation, we see that there are hypomethylated regions that correspond very nicely to the accessible chromatin regions, again, specific to the brain capillary endothelial cells, and also in the region that's being transcribed, there's hypomethylation. So this is really just a first step. We want to reconstruct the full pathway from the membrane to the transcriptional state of the cells and see how that relates to not just normal health, but disease, and not just in mice, but in humans. And so with that, I'll end it here and open the floor to any questions.
Jeremy, thank you very much for spending time with us and showing your data that spanned over a few decades, I guess, right? You started on that project quite long ago.
Yeah, 20 years.
My question to you is, can you speculate about the mechanism, in a kind of mechanistic way, what the function of GPR124 is in this complex?
Yeah, we don't fully understand it, I think it's fair to say. Our work has focused only on the role of two of the four extracellular domains, the most N-terminal two, which seem to be the business end in terms of interacting with Wnt7A and 7B and the other receptor complex proteins. But I think all we can say is operationally that when it's present, signaling is a lot higher, and it is specific to Wnt7A and Wnt7B. But we don't have any further insight than that.
Okay. Any other questions?
Hey, I have a question. Can you hear me?
Steve, yeah, good to see you.
Hi, good to see you too. I guess you're seeing my avatar because it's much better looking than I am.
You're still good looking.
Jeremy, very interesting. I was just wondering, for the frizzled proteins, these have these multi-transmembrane segments. How much of that protein do you need in the membrane portion to have a functional receptor?
That is a really good question. I think amazingly, 25 years after defining their role in Wnt signaling, that still is not clear. If you just express the extracellular domain as a GPI-anchored protein, it doesn't work. So you need something in the membrane, but we don't really know what you need and how much you need. There's a little bit in the C-terminal tail that's clearly important for mutagenesis studies, but the whole thing, I mean, it looks like a GPCR. It looks like the transmembrane regions should be doing something, but we don't know what they're doing.
Okay, that's really interesting. And the second thing, this is kind of trivial, but I think in one of your earlier slides where you showed, I think, the Noren knockout mouse retina, it looked like you had a section of the outer nuclear layer that was deformed.
Yes, oh, you're right, good eye. Yeah, you get folds in the retina.
So that's a reproducible thing where you get folds and possibly even, I don't know, like those rosette type things that occur sometimes in dysplasia.
Yeah, it's not as prominent as some of the more rosette-prone mouse lines, but you get a little bit of folding. I don't know if that's primary or secondary. Maybe there's, I don't know, some gliosis or traction on the retina or something. We're not sure.
Okay, I just noticed it was in the ONL but not in the INL. And I didn't know if it was just happened to be a weird section or whether that's a reproducible thing.
No, that's true.
Okay, thank you.
Yoshi.
Thank you very much. Can you hear me?
Yes.
Great. I'm Yoshi Manis from Indiana University. I have a question about the compromised blood-brain barrier in frizzled for knockout. Can you re-establish the blood-brain barrier if you reactivate the frizzled for in especially in adult or mouse?
Yes.
I'm very interested in pharmacological application, for example, delivering drug to the brain. So you can.
Yeah, we've done one experiment along those lines, and that is if you start with a Noren knockout mouse, in addition to the severe retinal disease, it has a mild decrease in barrier function in the cerebellum. It's not enough to kill the mouse, but you can easily see it with the leakage of the intravascular tracer. And so if you cross into that background a Noren cDNA that is under Cree control, you can activate it with the Cree ER trick later in life. And when you do that, the barrier in the cerebellum is restored within a week, I would say. We haven't done a very careful time course, but a week later, the barrier is normal in the cerebellum.
This is Corinne. I have a question. Can you hear me?
Yes, please. Go ahead.
So in the Noren blind people, since it's a familial genetic disease, can you eventually turn on the beta-catenin to rescue the phenotype in real people and humans?
Yeah, that's a good question. With, I'm afraid, a very inadequate answer. Those children are born blind, the ones with Nori disease. The development that is probably relevant happened in utero. We would have to, I think, be able to access them in utero to fix it. By the time they're born, the retina is so damaged by the gliotic response, I don't think there's any way to repair it. Now,
but if you did amniocentesis, like found out that they're going to have the disease from, in utero, because it's a familial disease, so we can check for it, right?
Yes, I mean, for
Possible?
those. Yeah,
Can
yes,
you
for
give it to
those.
the mom or
Yeah, I mean, if a family knows that there's the disease in the family and they want to do, prenatal diagnosis, of course, with the proviso that they would be aborting the fetus if it has the mutant allele, that's something that's here now. I mean, the one thing I'll add to that is that some fraction of the mutations are new mutations. There is no family history. So for those individuals, that's not an option. But just to,
You had a question?
Who was that?
Tim, did you have a question?
So you described differences between the vascularization and the vascular integrity, and as well permeability changes. To what extent are these related to each other? Does permeability affect capillary integrity?
yeah, I think what we can say is, the thing we can say that's most firm is at the level of gene expression. That is, we have from the transcriptomic analysis the list of genes and how much each of them changes with and without beta-catenin signaling, and also how they differ in CNS capillaries versus non-CNS capillaries. Clearly, it's a long list. It's in the hundreds. It includes the tight junction proteins. It includes the extrusion pumps, like the multi-drug resistant pumps, which basically are in brain capillaries and are there to pump toxins out. it includes all the transporters and so on. So I think at the level of gene control, it's a whole package of genes.
Thank you.
Anybody else?
Yes, Dorota here. First of all, wonderful talk and amazing images. I was wondering if GPR 124 and REC have other ways to work in organs that is not really rich in Wnt7A and B. And I think this is a case for retina itself.
Yes. Well, first, let's talk about REC for a second. REC is a multi-domain protein. It has, for example, protease inhibitory activity and protease inhibitory domains. And I think the evidence, this is from Noda's lab in Japan, the evidence is that it is a protease inhibitor, and that's part of its function. In fact, it plays a role in Notch signaling because it controls proteolysis that releases Notch ligands from the membrane. But for GPR, I think we don't really know. We're not seeing, we and others are not seeing a phenotype outside of the CNS. Maybe it's redundant with something, so it's not a phenotype that can be seen with a single mutant, but it certainly has other domains. It's a big protein, so maybe there are other functions.
Thank you.
Roxana, you had a question?
Yes, I have a question. I enjoy very much your talk. I have a question in regards of the blood-retina barrier. Is there any difference of this sort of mechanism of frizzle wind between the outer barrier versus inner barrier? It was quite interesting how the brain actually have those specific sort of labeling for various markers. Is there any difference between the outer versus inner?
Yeah, it's interesting. I'm glad you asked that because I took out a slide related to that in interest of time. But the intraretinal capillaries, as far as we see, are behaving just like capillaries in the brain and spinal cord, in that they have a barrier function normally, essentially zero free permeability, and it is under Wnt control. But if you look at the choriocapillaris, the barrier is the RPE, basically, because the capillaries are highly permeable. Actually, those capillaries are like the capillaries in the circumventricular organs. They're filled with fenestrations and have extremely high permeability. And in fact, when we stabilize beta-catenin in vascular endothelial cells, we convert the choriocapillaris to a blood-brain barrier type state. That is, we eliminate the fenestrations. So it is under, maybe I shouldn't say it's under Wnt control. It probably has an active mechanism to suppress Wnt signaling, to keep Wnt signaling down. Because if Wnt signaling went up, it would lose its permeability.
Thank you.
All right, if there are no more questions, again, I will see you a week from now at the same time, the same place. And Jeremy, thank you very much. And final word goes to Professor Brown, or he disappeared now. Then we'll go to Professor Kaiser.
I'm here.
Okay, final word goes to you.
That was a really fascinating talk, and the one thing that's got me thinking a little bit about is a very rare condition that we see clinically called Susac syndrome, which has a retinal vasculitis and hearing loss, with also some gray matter lesions in the brain. And I'm just wondering if this is the dysregulation of Wnt and beta-catenin signaling that occurs in focal areas randomly, but it can be controlled with steroids, so it seems like it might be an outlier to think about it that way.
That's interesting. Can you spell that for me? I don't know that one.
Susac, S-U-S-A-C.
Is it heritable?
No, it's idiopathic entirely, and it presents with focal areas of retinal arteritis with arterial occlusion, as well as hearing loss, and it can sometimes mimic multiple sclerosis, because like with Dawson's fingers radiating from the corpus callosum, you can have focal areas of infarction in the corpus callosum and kind of a little vacuole forming on the MRI. So this is I'm just wondering if this is a focal dysregulation, maybe multifactorial, but something worth thinking about a little bit.
That's interesting. Thank you. I'll look that up.
Thank you.
And if I call Professor Kaiser, then will be a final word from Professor Kaiser.
Thank you, Dr. Nathans, for a very stimulating talk. It's wonderful.
Phil, it's great to see you again. I want to just thank everyone, and I hope you all stay well. And it's been a pleasure and an honor to be here. I really appreciate it.
Thank you. Thank you so much.